Between the 4th and 5th week of your development, before your mother even knew what you looked like, you were growing a tail. 10 to 12 vertebrae, extending from the base of your forming spine. If someone had placed your embryo beside a mouse embryo at that stage, they would have struggled to tell the difference. By the 8th week, your tail was gone. The cells were broken down and reabsorbed back into the body, and all that remained was a small triangle of fused bone at the very bottom of your spine. You know it as your tailbone, or the coccyx. Most people never think about it unless they fall hard enough to bruise it. But the question it leaves behind is enormous. Something in your DNA actively shuts down tail development midway through. And whatever that switch is, it first appeared roughly 25 million years ago, when the ancestors of every living ape stopped growing tails for good. Gibbons, orangutans, gorillas, chimpanzees, bonobos, humans. An entire branch of the primate family tree, permanently tailless. For decades, nobody could explain what actually caused it. The strange part is that losing a tail should have been a terrible idea. Almost every other primate kept theirs, and for very good reason. Old world monkeys rely on their tails for balance, using them to correct posture mid-leap while sprinting along the tops of branches at full speed. New World monkeys in South America took it further, evolving prehensile tails powerful enough to grip branches and support their full body weight, a working fifth limb. From an engineering perspective, a tail is one of the most efficient stabilizing tools biology has ever produced. Giving one up is not an obvious improvement. It is surrendering a proven instrument. And every explanation scientists offered for why it happened ran into the same problem. They could not find the genetic mechanism behind it. Some argued tails became too expensive to maintain as body size grew. Others proposed they became unnecessary as movement patterns shifted. These ideas were reasonable, but none of them pointed to the specific molecular change that made tail loss happen. Without that, every theory was just a plausible guess. The answer came from a graduate student with a sore tailbone. In 2019, Bo Xia was a PhD candidate at NYU when a minor car accident left him with a coccyx injury. The recovery took close to a year. Sitting was painful. Walking was uncomfortable. And because this one small bone refused to stop reminding him it existed, Xia started thinking about it far more seriously than most people ever would. Why is this bone even here? Why do humans still have the remnant of a tail but not the tail itself? What exactly happened at the level of DNA to remove something that nearly every other vertebrate species on the planet still carries? According to his own account, he went home that same night and started reading everything published about tail loss in primates. Whether that qualifies as the most productive reaction to a car injury in scientific history or as evidence that graduate school permanently changes a person's priorities is an open question. Probably both. He started looking systematically. More than 100 genes had already been connected to tail development in various vertebrates. Using a genome browser, essentially a search engine for comparing genetic code across species, Xia and his colleagues began comparing those genes between tailed primates like macaques and baboons and tailless apes like chimps and gorillas. They were hunting for something specific to the ape genome, a difference that appeared exactly where tail loss showed up in the evolutionary timeline, and nowhere else. They found it inside a gene called TBXT. TBXT had been connected to tail development since 1927, when researchers first observed that mice carrying mutations in this gene grew abnormally short tails. The gene's other name, brachyury, literally means short tail in Greek. It plays a fundamental role in building the posterior body during embryonic development. What Xia's team discovered was a small piece of mobile DNA called an alloy element sitting inside one of the gene's introns. An intron is a region that does not directly code for protein, which is part of why nobody had looked there closely before. This insertion was present in humans, chimpanzees, gorillas, and every ape they checked. It was completely absent from monkeys. Allo elements are sometimes called jumping genes. They are short DNA sequences that can copy themselves and drop into new locations across the genome. The human genome carries over 1 million of them. Most do nothing measurable. For years, they were dismissed as junk DNA, biological clutter with no functional purpose. But this particular alloy had landed next to another, older allo element sitting in the reverse orientation. Together, the pair changed how the cell reads the TBXT gene. Instead of producing only the normal TBXT protein, the gene now also produces a shorter, altered version through a process called alternative splicing. The cellular machinery, encountering these two elements, skips over a section of the gene's instructions. The protein that results is structurally different, and that difference appears to be enough to shut down tail formation during embryonic development. One piece of mobile DNA, dropped into one intron of one gene, apparently rewired how an entire body part gets built. But finding the mechanism was only half the work. Proving it required something more concrete. Zia's team took this specific genetic change and engineered it into laboratory mice. Mice normally grow long tails. With the ape-like ALU-Y insertion placed into their version of the TBXT gene, some mice grew shortened tails. Others grew no tail at all. The outcome depended on the ratio of normal to altered protein being produced. A single genetic insertion, identified by a researcher who would never have started looking if his tailbone had healed faster, was enough to dramatically change tail development in a living animal. The study was published in Nature in February 2024. Then the experiment revealed something nobody had expected. Some of the mice also developed neural tube defects. The neural tube is the embryonic structure that becomes the spinal cord and brain. When it fails to close properly, the consequences are severe. Spina bifida, where the spinal column remains partially open, belongs to this category. This meant the genetic change that removed tails from the ape lineage did not arrive cleanly. It came packaged with a measurable increase in the risk of a serious birth defect. Not a simple upgrade, a trade-off written into every ape genome for the last 25 million years. And this is not ancient history. Neural tube defects still affect roughly 1 in every 1,000 human births today. It is one of the reasons doctors push folic acid supplementation so strongly during early pregnancy, because folate supports proper neural tube closure. The idea that a genetic insertion from 25 million years ago might still be quietly nudging up that risk in modern pregnancies is deeply uncomfortable to sit with, which forces a harder question. If losing the tail came with a genuine cost, paid in every generation since, why did natural selection keep it? What was so valuable about being tailless that it outweighed an increased risk of spinal birth defects across millions of years? The answer is in how apes moved. Around the same time tail loss appeared, ancestral apes were changing their locomotion. Monkeys run along the tops of branches on all fours. Their tails serve as counterweights, correcting balance during sprints and stabilizing their bodies mid-leap. For that style of movement, a tail is essential hardware. Apes began doing something different. They started swinging beneath branches, arm over arm, in a style called suspensory locomotion. Gibbons still move this way today, covering enormous distances through forest canopies at startling speed, their whole body suspended from their hands. When you are hanging below a branch, instead of running along the top, a tail stops being a counterweight. It becomes dead weight. Extra mass to carry, extra length to catch on branches with no stabilizing function left to justify the cost. Losing it would have streamlined a body already reorganizing itself around shoulder mobility, grip strength, and vertical climbing. And the consequences did not stop in the canopy. A torso built for hanging vertically, with shoulders designed for overhead reach rather than four-legged running, is structurally closer to a body that can stand upright. Many researchers believe suspensory locomotion laid the foundation for bipedalism in the human lineage. The same body plan that made swinging through trees efficient eventually made walking on two legs possible. So the chain runs like this. A piece of mobile DNA landed in the wrong intron of a single gene, somewhere in Africa, in an animal that had no idea anything had changed. That insertion altered how the gene produced its protein. The altered protein disrupted tail formation. Without a tail, a new style of movement through the canopy became more efficient. That movement reshaped the ape body toward vertical posture, and vertical posture, over millions of years, became walking upright on two legs, with two hands permanently free for tool use, complex manipulation, and eventually building everything you see around you today. 25 million years of consequences started by a piece of DNA that landed in a spot it was never aimed at. There is one final detail. Every so often, very rarely, a human baby is born with a small soft tissue protrusion at the base of the spine. Doctors call it a vestigial tail. It typically contains no vertebrae and is removed with straightforward surgery. But each case is a quiet signal that the instructions for building a tail were never deleted from the human genome. They are still there, buried in the code, mostly silenced by the developmental override that one Louis insertion put in place. Your tailbone is the last visible remnant of a body part your ancestors carried for hundreds of millions of years. Shut down by a single piece of wandering DNA that arrived with a hidden cost and was kept anyway, because what it made possible was worth the price.